Okay, we'll finish up with this slide and then move on. Generally, and I think I maybe didn't quite say it accurately enough, there can be a shift from intracellular fluid to extracellular fluid, but the mechanisms that we are talking about with regard to hormonal or response from the kidneys or the respiratory system or even buffer systems, they are in response to what's going on in the extracellular fluids. So the regulatory response would be extracellular in the extracellular fluid in makeup, but certainly has an effect on the intercellular fluid because the intercellular and extracellular fluid in that immediate area is in equilibrium. So if something is dramatically off with the intracellular fluid, you're either going to have fluids shift from one area to the other because from intracellular to extracellular, or vice versa, because the two of those together are generally in equilibrium. If in fact there's a need for a shift, that shift will be rapid, but will reach equilibrium again in a few minutes to a few hours. So that there is movement between the two compartments, intercellular and extracellular, but it happens on usually a very local level, and those shifts then can be reach equilibrium very fast. But understand that if in fact you have a shift from the extracellular to the intracellular, you're going to set up a scenario where you have dehydration, and the whole process of ADH, increasing thirst and water being taken in and water being spared by the kidneys is going to take place, but that's in response to the extracellular fluid loss, which was lost because the cells inside need water. The intracellular fluid tends to be a fluid storage compartment. That's where fluids are stored. So usually what happens is it goes from intracellular to extracellular. And then there can be quite a fast shift if need be, but those regulatory processes that we talked about, hormonal, whether kidney or respiratory, they take a little bit longer to reach equilibrium than minutes and hours. The problem with there being a dramatic shift between fluid compartments, intracellular, extracellular, is this idea of hypertonic, hypotonic. You learned a little bit about this in 141. So the number of solute equivalents in water needs to remain the same whether it's intracellular or extracellular. Now please understand that you have a larger water compartment inside the cell, but you also have more solutes than outside the cell. And if in fact outside the cell you suddenly have water loss, now the solutes are more concentrated. They're more concentrated outside the cell than inside the cell in that immediate area. And so what the outside the cell, the extracellular volume, extracellular fluid is hypertonic with respect to the inside the cell. And that will cause water to move from inside the cell to the outside of the cell. We call that a fluid shift. And this is what happens the other way, actually, if you overhydrate somebody, or if somebody is dehydrated, I guess, but clinically, if you're putting an IV in, you're putting fluids in, this is what you are trying to do. You're trying to hydrate that extracellular fluid, so in fact, that the intracellular fluid then between the intracellular and the extracellular fluid, you can reach equilibrium. Cells need water to work, so that equilibrium has to be met and kept in order for normal metabolic behaviors to take place. The cations that are most important are sodium and potassium. They're most important for the functioning of the cells, whether of the cell, whether it's inside the cell or outside the cell. And they're the biggest contributors to osmotic concentrations in a huge way. So you have extracellular, you have sodium being the extracellular cation, that again is the biggest amount of solutes is sodium. And so if sodium is out of balance, you have problems. The balance is based on several factors. Is the uptake of in the digestive system of sodium ions. So how many are you taking in digestively, and then how many are you secreting in the kidney? And what happens is typically the gain and loss each day, but it's you're able to keep the gain and loss in equilibrium. If you have too much loss of sodium, you have hyponatremia, and this can be because of increased GI loss of sodium. This could be for various reasons in the GI. And I really apologize for the slide. It's kind of bad, but it does have some pretty good information on it. So dietary intake of sodium is reduced dramatically. You have excessive water intake, excessive phosphorylation, so metabolic behavior, decreased GI loss of water, and then with hypernatremia, so too much sodium, you have too much, and that tends to be more of a problem that we have. You have increased intake in sodium, and decreased urinary loss of sodium. So both of those are potentially dangerous because of the need for sodium out in the extracellular fluid to maintain that osmotic concentration, the number of solutes in the water. And also sodium is very important for a nerve impulse. In terms of, this one's a little better than the other one. In terms of potassium, remember potassium is K+, that's inside the cell. You have decreased GI loss, if with hyperkalemia you have excessive intake, decreased urinary secretion, decreased GI loss. Actually decreased sweating, I guess you might say. That's the fancy way of seeing decreased transcutaneous loss, decreased sweating. Or with hypokalemia, poor intake, increased urinary secretion, and then increased sweating with that. Both are potentially dangerous, not only for osmotic concentration, changes in pH, and then also aldosterone levels, or you have hormones that come in and you have regulations for each of these, as long as the body is given enough time to regulate both of them. Types of acids in the body: you have volatile acids, you have fixed acids, and you have organic acids. Volatile acids tend to be those that you can breathe out, like carbonic acid. Volatile acid is created by interaction with carbon dioxide, and you can breathe it out. It's a formula where carbon dioxide and water gets together to form carbonic acid and a bicarbonate ion, and that equation moves back and forth. Organic acids are a byproduct of your metabolic behaviors, like the breakdown of the production of lactic acid. If you're using fats or proteins in the absence of oxygen, lactic acid, or if you are the ketone bodies from synthesis of acetyl-CoA, using fats to make acetyl-CoA. Fixed acids are acids that don't leave solution once they are produced. They remain in body fluids. They have to be eliminated by the kidneys. So three types of acids. But just to review, an acid is anything that causes pH to go skew more towards the lower range. Bases are anything that causes pH to skew more toward the higher range. I guess I'll stop there. And then because blood needs to stay within a certain range, you have buffer systems. And buffer systems tend to operate either intracellular or extracellular. And dramatic changes in hydrogen ion concentrations are basically dangerous for you. Your brain doesn't like changes in the pH of blood. Three major chemical buffer systems are the phosphate buffer system, and that is the important one in the intercellular fluid. Protein buffer systems are important intercellular and extracellular, and protein buffer systems can include hemoglobin, which actually can carry carbonic acid, amino acid buffers, and then also plasma protein buffers. Extracellular fluid exclusively is the carbonic acid bicarbonate buffer system, is the most important in the extracellular fluid. And here you have the definition of what pH of blood should be, should remain 7.35 to 7.45. Above 7.45 you have alkalosis. Below 7.35 you have acidosis. And ions and for example, ketone bodies the fixed and organic acids and even the volatile acids are created in part of the metabolic processes. So most of that base, that acid-base balance is maintained by the kidney because you can either excrete out hydrogen ions or retain them. But locally, these buffer systems work to make sure that pH stays within a normal range in very controlled areas. So the fast acting, the immediate acting is going to be these local blood buffer systems. And carbonic acid is one of the most, remember, it's the most used in the extracellular fluid, and that one is the one that basically the body monitors more so than the intercellular fluid. And what happens is if you go if you're going towards breathing out CO2, so you're looking at the bottom here, the red arrows at the bottom, is you have a bicarbonate reserve, and that is with a breakdown of sodium. Sodium bicarbonate breaks down into bicarbonate ions. Bicarbonate ions can get together with those extra hydrogens. That's if you have an increase in acidity of the blood. That creates carbonic acid. Carbonic acid breaks down into carbon dioxide and water, which is exactly what you breathe out. If you have, you also take it, you have an increase, so you have something that's basic, then what you're going to actually do is take that CO2 that's floating around in the blood, mix it with water, create carbonic acid, put that together with hydrogen, and turn a bicarbonate ion and turn that into sodium bicarbonate. So the sodium bicarbonate there at the end is your reserve. It can go either way. You can have that reserve of it, or you can break it down and then breathe it out. And we'll pick this up on the next one.